अस्सलाम वालेकुम स्टूडेंट्स, होप यू ऑल आर फाइन। स्टूडेंट्स, आज हम बायो क्लास 11 चैप्टर नंबर 6 एक्सरसाइज लॉन्ग क्वेश्चन आंसर्स सॉल्व करेंगे। सो लेट्स स्टार्ट। फर्स्ट क्वेश्चन है हमारे पास, एक्सप्लोर द टेक्सोनोमिक एंड फोटोसिंथेटिक पोजीशन ऑफ प्रोकैरियोट्स। Prokaryotes are a group of microorganisms that lack a true nucleus and other membrane-bound organelles. They are divided into two domains, archaea and bacteria. Taxonomic position. Domain archaea. Phylum Euryarchaeota, methanogens, celophiles. Phylum Crinaarchaeota, thermophiles. Phylum Proteobacteria, some species. Domain bacteria. Phylum Proteobacteria, most species. Phylum Firmicutes, gram-positive bacteria. Phylum Actinobacteria, gram-positive bacteria. Phylum Cyanobacteria, photosynthetic bacteria. Photosynthetic position. Only cyanobacteria within domain bacteria are photosynthetic prokaryotes. They contain thylakoids where photosynthesis takes place. They produce oxygen as a byproduct of photosynthesis. They are thought to be the ancestors of chloroplast in eukaryotic cell. Other prokaryotes. Most prokaryotes are non-photosynthetic and obtain energy through respiration, aerobic or anaerobic fermentation, chemolithotrophy, using inorganic compounds as energy source. In summary, prokaryotes are divided into two domains, archaea and bacteria, with only cyanobacteria within domain bacteria being photosynthetic. The rest of prokaryotes rely on other energy producing mechanisms. Next question is, rationalize the universal presence of bacteria across diverse habitats. The universal presence of bacteria across diverse habitats can be rationalized by considering their ancient origin. Bacteria evolved over 3.5 billion years ago, allowing them to adapt to various environments. Small size. Bacteria's tiny size enables them to thrive in microscopic niches and survive in extreme conditions. Simple nutritional requirement. Bacteria can grow on minimal nutrients, making them capable of surviving in nutrient-poor environment. Metabolic versatility. Bacteria have diverse metabolic pathways, allowing them to exploit various energy sources and thrive in different environments. Ability to form biofilms. Bacteria can form protective biofilms, enabling them to withstand harsh conditions and colonize new habitat. High reproductive rate. Bacteria's rapid reproduction allows them to quickly adapt to changing environment and colonize new areas. Horizontal gene transfer. Bacteria can share genes with each other, facilitating the spread of adaptations and increasing their ability to thrive in diverse habitat. Symbiotic relationship. Bacteria form symbiotic relationship with other organisms, expanding their habitat range and increasing their access to resources. Next is resistance to extreme conditions. Bacteria have developed mechanisms to survive extreme temperatures, pressures, salinity, and radiation. Evolutionary adaptability. Bacteria's ability to evolve rapidly enables them to respond to changing environments and exploit new habitats. These factors combined have enabled bacteria to colonize almost every habitat on Earth, from the human gut to deep sea vents and from freezing tundra to hot springs. Next question is, differentiate between conjugation, transformation, and transduction in bacteria elucidating their respective functions. First of all, we see conjugation. Function: direct transfer of genetic material, plasmid or chromosomal DNA between two bacterial cells through a specialized structure called a pilus. Process: donor cell F positive produces a pilus, recipient cell F negative attaches to the pilus, genetic material is transferred from donor to recipient. Result: the recipient cell becomes genetically modified, acquiring new traits. Transformation function: uptake of free DNA molecules from environment, allowing bacteria to acquire new genetic traits. Process: bacteria release DNA into environment through lysis or secretion. Competent bacteria take up free DNA molecule. The acquired DNA is integrated into bacterial genome. Result: the transformed bacteria exhibit new characteristics. Transduction function: transfer of genetic material from one bacterium to another through a bacteriophage virus which infects bacteria. Process: a bacteriophage infects a donor bacterium. The phage packages bacterial DNA along with its own genetic material. The phage infects a recipient bacterium, transferring the bacterial DNA. Result: the recipient bacterium acquires new genetic traits. In summary, conjugation: direct cell-to-cell transfer of genetic material. Transformation: uptake of free DNA molecules from environment. Transduction: transfer of genetic material through a bacteriophage. These mechanisms enable bacteria to share genes, increasing genetic diversity and facilitating adaptation to changing environments. Next question is, provide an overview of bacteria's morphology, dimensions, and structural characteristics. Morphology: spherical or cocci, staphylococcus, streptococcus, heart-shaped bacteria which are called bacilli. Example is Escherichia coli, Bacillus species, spiral, Spirilla. Examples are Helicobacter, Spirillum, comma-shaped Vibrio. Example is Vibrio cholerae, pleomorphic irregular shapes, for example mycoplasma. Dimensions: length 0.5 to 10 micrometers, width 0.2 to 2 micrometer, diameter spherical bacteria 0.5 to 2 micrometer. Structural characteristics: cell wall. Peptidoglycan, also known as murine layers, provide rigidity and maintain shape. Teichoic acids and lipoteichoic acids present in gram-positive bacteria. Cell membrane. Phospholipid bilayer with embedded protein, selectively permeable, controlling flow of nutrients and waste. Capsule. Outer layer of polysaccharides or polypeptides, protects against desiccation, phagocytosis, and antibiotics. Flagella. Long whip-like structure for motility, composed of flagellin protein. Pili. Short hair-like structures for attachment and DNA transfer, composed of pillin protein. Ribosomes. Small organelles responsible for protein synthesis, found throughout the cytoplasm nucleoid region containing the bacterial chromosome, lack a true nucleus. Inclusions: storage organelles, granules for nutrients, waste, or other substances. Examples: glycogen, polyphosphate, and sulfur granules. These structural characteristics enable bacteria to maintain their shape, regulate their internal environment, and interact with their surroundings. Next question is, elaborate on the significant economic contributions of bacteria. Bacteria make significant economic contributions in various industries including food and beverage, fermentation, yogurt, cheese, bread, beer, wine, soy sauce production, food preservation, pickling, salting, and curing. Nutritional supplements, probiotics, and vitamins. Next is pharmaceuticals, antibiotic production like penicillin, streptomycin, and tetracycline, vaccine development using bacterial antigens or vectors, drug delivery, bacterial vectors for gene therapy, agriculture, nitrogen fixation, rhizobia bacteria in legume nodules, biofertilizers, phosphorus solubilization and micronutrient supply, biopesticides, bacterial insecticides and fungicides, biotechnology, genetic engineering, bacterial expression systems for protein production, bioremediation, cleaning pollutants using bacterial degradation, biofuel production, bacterial fermentation for ethanol and butanol, environmental services, wastewater treatment, bacterial degradation of organic matter, biodegradation, oil spill cleanup and plastic degradation, climate change mitigation, bacterial carbon sequestration. Next is mining and metallurgy, bioleaching, bacterial extraction of metals from ores, biomineralization, bacterial formation of minerals and ores, cosmetics and personal care, skincare, bacterial fermentation for hyaluronic acid and glycerol, hair care, bacterial enzymes for hair straightening and smoothing, animal health, probiotics, bacterial supplements for animal nutrition and health, vaccines, bacterial antigens for animal vaccines. These economic contributions highlight the significant impact of bacteria on various industries and our daily lives. Next question is, present a comprehensive account of archaea, a distinct domain of microorganisms. Archaea is a distinct domain of microorganisms that are prokaryotic, meaning they lack a true nucleus and other membrane-bound organelles. They are thought to be one of the most ancient forms of life on Earth, with fossil evidence dating back over 3.5 billion years. Characteristics: first one is cell wall. Archaea have unique cell wall composed of ether lipids, pseudopeptidoglycan or glycoproteins. Metabolism. Archaea exhibit diverse metabolic processes including methanogenesis, halophily, thermophily, and chemolithotrophy. Genetics. Archaea have distinct genetic features such as the presence of introns and oprons. Replication. Archaea replicate using a unique replication system involving the enzyme DNA polymerase B. Types of archaea: methanogens produce methane gas as a byproduct of metabolism. Halophiles thrive in extremely salty environment. Thermophiles grow optimally at high temperature above 45 degrees Celsius. Psychrophiles grow optimally at low temperature below 15 degrees Celsius. Chemolithotrophs derive energy from inorganic compounds. Habitats: extreme environments like hot springs, salt lakes, deep sea vents, human gut. Some archaea are found in human gut microbiome, soil and sediments. Archaea are present in soil and sediment ecosystems. Importance: ecological roles. Archaea play crucial roles in nutrient cycling, decomposition, and greenhouse gas production. Biotechnological applications. Archaea are used in bioremediation, biofuel production, and enzyme manufacturing. Evolutionary significance. Archaea provide insights into the early evolution of life on Earth. In summary, Archaea are a fascinating group of microorganisms that have adapted to thrive in diverse environments from extreme ecosystem to human gut. Their unique characteristics and metabolic processes make them an important area of study in microbiology, ecology, and biotechnology. Next question is, Explain the application of bacteria in both research and technology. Bacteria have numerous applications in both research and technology, including research. Genetic engineering. Bacteria are used as vectors for gene transfer and expression. Molecular biology. Bacteria are used to study gene function, regulation, and expression. Cell biology. Bacteria are used to study cellular processes such as cell division and signaling. Evolutionary biology. Bacteria are used to study evolutionary processes such as adaptation and speciation. Environmental science. Bacteria are used to study environmental processes such as nutrient cycling and biodegradation. Next is in technology. First is biotechnology. Bacteria are used to produce biofuels, bioproducts, and pharmaceuticals. Food industry. Bacteria are used in food production such as yogurt, cheese, and bread. Agriculture. Bacteria are used as biofertilizers, biopesticides, and biostimulants. Environmental remediation. Bacteria are used to clean up pollutants such as oil spills and toxic chemicals. Medicine. Bacteria are used to develop vaccines, diagnostics, and therapies. Synthetic biology. Bacteria are used to design and construct new biological systems. Next is bioremediation. Bacteria are used to clean up contaminated soil and groundwater. Bioenergy. Bacteria are used to produce biofuels such as ethanol and butanol. Other applications. Bioreactors. Bacteria are used in bioreactors to produce biofuels, bioproducts, and pharmaceuticals. Biosensors. Bacteria are used to detect environmental pollutants and toxins. Biocatalysts. Bacteria are used as catalysts for chemical reaction. Probiotics. Bacteria are used as probiotics to promote human health. In summary, bacteria have a wide range of application in both research and technology, from basic scientific research to practical applications in industry, medicine, and environmental remediation. Next question is, investigate the remarkable diversity in bacterial nutrition strategies. Bacteria exhibit a remarkable diversity in their nutrition strategies which enable them to thrive in various environments. Here are some examples. Autotrophy. Some bacteria, like cyanobacteria, produce their own food through photosynthesis or chemosynthesis. Heterotrophy. Most bacteria obtain nutrients by breaking down organic matter such as sugars, amino acids, or other macromolecules. Saprotrophy. Bacteria like Pseudomonas and Bacillus decompose dead organic matter, recycling nutrients. Parasitism. Certain bacteria like Mycoplasma depend on host cell for nutrients. Symbiosis. Bacteria like Rhizobia form mutualistic relationship with plants, providing nitrogen fixation in exchange for carbohydrates. Next is chemosynthesis. Bacteria like hydrogen bacter and nitrobacter oxidize inorganic compounds generating energy. Lithotrophy. Bacteria like Thiobacillus and Ferrobacillus use inorganic substances as energy sources. Mixotrophy. Some bacteria like Chlamydomonas combine autotrophic and heterotrophic nutrition strategies. Oligotrophy. Bacteria like Colobacter and Hyphomicrobium thrive in nutrient-poor environments. Carpiotrophy. Bacteria like E. coli and Bacillus subtilis grow rapidly in nutrient-rich environment. This diversity in bacterial nutrition strategies allows them to occupy a wide range of ecological niches from human gut to deep sea vents and from soil to extreme environments. Next question is, contemplate how the absence of bacteria might have altered life on Earth. The absence of bacteria would have profoundly altered life on Earth, leading to no decomposition, organic matter would accumulate and nutrients wouldn't be recycled, impacting plant growth, disrupted nutrient cycles. No nitrogen fixation, phosphorus solubilization, or sulfur reduction, affecting plant and animal nutrition. No fermentation, no yogurt, cheese, bread, beer, or wine production, impaired human health. No gut microbiome, leading to compromised immune system, digestion, and overall well-being. Reduced agricultural productivity. No bacterial fertilizers, pest control, or plant growth promoters. Next is altered ecosystem. Changes in soil structure, water quality, and atmospheric composition. No antibiotics. Bacteria are a primary source of antibiotics which would be absent. Impacted climate. Bacteria influence greenhouse gas emissions and their absence would alter Earth's climate. No bioremediation. Bacteria clean pollutants. Without them, environmental damage would increase. Alternative evolutionary paths. Life on Earth might have followed a different trajectory, potentially leading to unfamiliar forms of life. In summary, the absence of bacteria would have significantly impacted various aspects of life on Earth, from ecosystem and human health to agriculture and climate. Their presence is vital for maintaining the balance and diversity of life as we know it. Next question is, analyze the key roles played by prokaryotes in the biosphere and human society. Prokaryotes, including bacteria and archaea, play crucial roles in biosphere and human society. First of all, we see in biosphere. Ecological balance. Prokaryotes maintain ecosystem balance through decomposition, nutrient cycling, and symbiotic relationships. Primary production. Cyanobacteria and other prokaryotes contribute to primary production through photosynthesis, decomposition, and nutrient cycling. Prokaryotes break down organic matter, releasing nutrients for other organisms. Soil formation and fertility. Prokaryotes influence soil structure and fertility through nitrogen fixation and other processes. Next is in human society. Food production. Prokaryotes are used in food processing, for example yogurt, cheese, bread, and as biofertilizers and biopesticides. Medicine. Prokaryotes provide antibiotics, vaccines, and insights into human health and disease. Biotechnology. Prokaryotes are used in biofuel production, bioremediation, and development of new products. Environmental monitoring and remediation. Prokaryotes serve as indicators of environmental health and are used to clean pollutants. Research and development. Prokaryotes are model organisms for scientific research, driving advances in fields like genetics and biotechnology. In summary, prokaryotes play vital roles in maintaining ecological balance, supporting human life, and driving innovation in various fields. Their contributions are essential for well-being of both biosphere and human society. So students, हमारे क्वेश्चंस कंप्लीट हो चुके हैं. होपफुली आपको समझ आ गए होंगे. If you have any doubt, you can ask in comment section. Please like, share and subscribe my YouTube channel. Thank you.